My talks throughout today are structured in like a triphasic manner. The first, we're just going to kind of go over precision medicine, what it is, what it can do, where it's going. The second talk will be a little bit more about the test itself and Aristotle as a service. And then the last talk is going to be looking at the science behind some of the breast cancer profiling that we do. So there is a Q&A session at the end, but feel free to ask whenever something comes to mind. It's a very intimate group, so we can stop and discuss things as they come up. What is this? Okay, so yeah, you know a little bit about us. You know, we co-founded this in our grad school days, and the goal was to do deep molecular profiling on individuals, from the nuclear DNA down through the expression of metabolites and dynamic changes of those metabolites, and everything in between. So we are launching a microbiome test at the end of the summer, hopefully, and Micah keeps pushing it back. And we are planning to launch a very large-scale proteomics panel, which will give you the markers you're traditionally aware of, like cholesterol, HbA1c, ALT, AST, and then go all the way up through to the transcriptome and genome. You've met Greg, some people on here that you may have not met. Denver is our precision health consultant as well, so you may consult with Denver on a few of these reports. Amy's our customer service rep. Carmen, we went to grad school with as well, and she's one of our consultants. Professor Klein Sitharaman, she's unfortunately in Copenhagen at a conference, so we cannot meet her today. But she was my PhD advisor. And Dr. Wisner is Alex's PhD advisor, postdoc advisor currently. And so they're on the third floor. We'll go up there. I'm not sure. Corey's traveling to ASN, isn't she? So she's in like Chicago or something, and Judith's in Copenhagen, as professors usually are. But they have been really instrumental in helping shape the overall direction and scientific trajectory of the company. Hey, Vishal. Hey. Thanks for making it, yeah. So that's, in a nutshell, who we are and what we do. And so our goals are, as a collective of scientists and physicians, is to bring to you these tools that are largely and historically been sequestered at research institutions, right? You'll see a chemistry autoanalyzer. Those are in hospitals and clinics, but a mass spectrometer is not. Illumina for whole genome sequencing is not. LC-MS, MS proteomics for proteomic and transcriptomic analysis is not available. And so these analyses are also not available, right? Usually you're stuck with a physician interpreting it based on their knowledge and experience, which is great. But when we start to get really into the models, we'll discuss how the brain really cannot compute that. So there's obviously some things in personalized medicine that are sort of like the hallmarks, right? We're able to tailor treatments. We're able to identify diseases early. When we do that, we increase survival rates. We improve prognosis. We're able to stratify patients based on various molecular characteristics and reduce adverse drug reactions. Ultimately, this leads to cost-effective health care. As Robert pointed out, that might not be the goal for some parties in this scheme. Empowering patients by giving them translatable, digested, coherent, and non-overwhelming health information. And then, of course, when we collect all of this data, especially as we get to multi-omic data, longitudinally sampled, we are able to lead research, as we'll see with one of our recent publications as well. And so on the right, you see sort of the dissonance and the challenge that's inherent in the data types that we are trying to assimilate. There are essentially 3 levels of these reports that we are going to go through, right? So we first have to take all these omics layers. We have to distill them, make sense of them, correct for differences in dimensionality. We then go through various analyses to select the features that are important, or maybe biologically relevant, a gene, its protein, and its downstream metabolite all operating within one metabolon. And then how do we take that into interpretable, useful, reliable, repeatable health metrics, like our aging index or our environmental toxicity score? And so this was actually a part of the paper that was published last night, was accepted last night, so you can read that paper and see this figure and some of our philosophy around it. So let's talk a little bit about the evolution of medical practice, right? So we have ancient medicine. This was like herbal healing, acupuncture, some spiritual healing. Of course, we have Hippocrates, Some bioethics emerges with the Hippocratic Oath. But really what we're, like the predominant theory at this time was humoral health, right? So for 3,500 years, what we had was this really unscientific, kind of, sort of intuitive, sometimes right, often wrong approach to medicine. And then for the next 2,000 years, we have medicine in the medieval ages, or the Middle Ages. And so this is really related to Europe, I will say, because medicine in the Middle Ages in the Middle East was far different. They were doing, Ibn Sina was doing glaucoma surgery in Shiraz, while barbers were doing surgeries in Europe. So we're talking about Western medicine here. And really what we had here were classical texts, but also religious beliefs. Like mental health was still demonic possession at this time. We do have some key developments here. So we have the first hospitals emerging. We have actual institutions that are training physicians, and the field is differentiating. So a physician is not a butcher anymore. Physicians often were other occupations. They weren't just dedicated physicians. And so now we're having really things that we're looking at is disease causation. People are thinking about what relies on what. But basically, the treatments at this age were predominantly bloodletting and leeches. And then we have Renaissance medicine, right? So now we're starting to get some renewed interests in anatomy. It is now a little bit less taboo to open up the human body and look inside. And so now we're going from 3,500 year span to 2,000 years to now a couple hundred years. And we're really getting, because of course, Andreas Vesalius, really kind of revolutionized and brought to the forefront of consciousness that anatomy, or the form of the body, will ultimately have some influence on its function. And so we're starting to dissect, we're starting to learn more about human physiology. And then we can go to medicine in the 19th century. So now we're at a 100-year iteration. We're no longer waiting 3,500 years for the next jump. We're at 100 years. And now we're looking at germ theory, right? Louis Pasteur, Robert Koch. Advances in microscopy now means that we can look at things and see that there are small organisms that change health. Antiseptics and vaccinations become a thing here. And we start to move toward evidence-based medicine. This is still not a scientific method. It is really engineering principles applied to medicine. And we start to ask ourselves about causality. What are the principles of causality, and how do we show these things to be true? Now, from 1900 to 2000, we're looking at 20th century medicine. Now we have x-rays. We just got antibiotics. And now we're getting advances in surgical techniques as well. Watson and Crick uncovered the structure, or I should say articulated it. Somebody else uncovered it in 1953. And we're starting to make advances with the vaccines. Our public health and epidemiology efforts are really paying off. And we've eradicated diseases like smallpox, right? And now we are in this period, the dawn of personalized medicine, precision medicine. In 2003, the Human Genome Project completes. And now we can look at specific genetic profiles. We can look at omics layers profiles. We're looking at the effects of environment on the lifestyle. We have fully adopted a scientific rigor behind this. And what we are really looking at now is big data, AI, and multi-omics. And I think the historians of science and medicine, the philosophers of science and medicine, they will look back on this time period and say this was a really special time. I think in a few hundred years, we will look back in this post-2003 era and think that, for the 50 years after that, we gained exponential, we were the benefactors of an exponential increase in knowledge, analytical throughput, and really taking the complexity off of just the physician interpreting this and putting it into these fancy algorithms and computational techniques. So precision medicine, of course, we know it's tailored to individual profiles. We call this figure the multi-omic man. You might see the multi-omic man appear in a few other slides throughout the day. We are trying to tailor these healthcare strategies. It is based on genetic information, proteomic information, in our case, metabolomic information. And we're going from this one-size-fits-all approach, which we don't utilize for our clothing, but we, for some reason, have assumed it was fitting for our healthcare, to customized treatments. We're going from reactive medicine to proactive medicine. And this is really important because we can improve the efficacy of our treatments by targeting specific things. When we make a recommendation for alpha lipoic acid in an individual, when we look at their mitochondrial health, that is because we have metabolites showing their rates of beta oxidation through the mitochondria are diminished, right? If we are giving them malic acid, we might think that their fatty acid biogenesis is diminished. So we'll feed them some more malic acid to increase that, to improve lipid membrane health. And then, of course, the enhanced patient experience. I think, as we'll get into later on, the patient really does care about testing. Testing, the perception of care and how many tests are done are significantly associated. We've seen this in study after study. And so the perception and the patient involvement, which is what patients want these days. They want to be involved in their care. This is what personalized and precision health allow us. And they are two different things, personalized and precision. We are talking about precision health today. And so we are going to sort of explore in our talks today how through advanced data analytics, advanced data collection through platforms like mass spectrometry and AI, we're able to yield some very interesting results. Why do I say that we need this AI? So here's the number of variables on the y-axis, and here's the number of unique models that you can make with those variables, right? When we get to 126, we're already at 10 to the 7 models being made. So we can make 10 million, models, unique models, each one looking at a different thing. One is looking at atherosclerosis, one is looking at atherosclerosis in the context of a high-fat diet. That would be two different unique models. So we can make 10 million models from our 126. We currently model about 15,400, give or take a few dozen. And when we get up to 25,000 models, that number becomes 10 trillion. So when we start talking about whole genome sequencing, large-scale proteomics, this is not something that Robert, as brilliant as you are going to be able to sit down and make sense of. We need to have data infrastructure and algorithms in place to give a first-pass analysis and at least flag things that your brilliant mind can then review, interrogate, and adjust based upon what you know of the patient that we don't know. So we need AI. So here's the systems biology pyramid. The definition of precision medicine is given here. We use precision medicine because personalized medicine really means that it's to that person only. That comes when we get to genetic data. That is personalized medicine. Precision medicine happens when you have data that cannot be used to uniquely identify an individual. So really you need either a bunch of data on very rare variants that only this person has or a whole genome sequence to talk about personalized medicine. So we're at precision medicine right now. And so we are looking at everything from the genome down through the metabolome, right? So we have transcription layer here from the genome to the transcriptome. These RNAs are being produced. Those RNAs will then go to the endoplasmic reticulum. If the endoplasmic reticulum is functioning fine, those will be correctly folded to functional proteins. The protein is the functional unit of life. And then from the protein, we have interactions with the microbiome. This is included for ease of understanding in the pyramid, but as Alex will point out, this should be its own layer. And every species is its own pyramid. So that's really how complex the microbiome is. And so currently it is, again, one of those black box things. Then we have the metabolome. And what we see here is that the metabolome is downstream of the genome, immediately upstream of the environment. It is that layer that contains all of the information we need to probe to look at gene-environment interactions. And most disorders and diseases are not genetic diseases, right? Huntington is, right? If you have the gene for Huntington's, you're going to develop Huntington's and die of it. The things that we die most from, the greatest causes of human morbidity and mortality, are things like heart disease. There are things like cancer. There are things like neurodegenerative disorders. And those are not relegated to just a few dozen even genes, right? If you have ApoE44 allele, you're going to have a higher risk of early onset Alzheimer's. But it doesn't mean that you're going to express that, right? So it is a product of gene-environment interactions. And so to understand these diseases and typify them more accurately, we need to probe that layer that contains the most sensitive and specific information, that layer that sits at the nexus of genes and environments. So, right, we named our test after Aristotle. Aristotle in the Nicomachean Ethics, knowing yourself is the beginning of all wisdom. And Aristotle was the first thinker to really commit a cogent theory of metabolism to writing. Aristotle, in the Metaphysis, talks about metabolism as being this thing that happens when you consume food that contained the building blocks, and then from food, you also get, some heat. So even he had like some sort of concept of entropy and heat generation in metabolism in his writings. And so we named it after him, as he is technically the father of metabolism. And metabolomics is used in various things. So if you look at, for instance, my own publication record, you will see something from all of these little aspects, right? You'll see clinical research, you'll see genetic variant screening in my work, chemical analysis, tox screening. We do behavioral interventions for, like let's say it's a sit-stand study, and we're looking at the effects of forced stand times during the workday. How does that change the metabolism? That's a behavioral intervention. Most of my work lies here in biomarker discovery, but metabolomics as a science, when it was first introduced by Dave Wishart in summer of 1998, since then it has enjoyed an exponential increase in interest, and as you can see, it is central to so many highly relevant niches of science. Metabolomics is the study of small molecules. These are less than 1,500 Daltons. So sometimes I get questions, can you look at cholesterol? Can you look at HbA1c? The answer is no, they're too big. They're far too big for the metabolomics platform that we have. We need another platform to look at those things. We are looking at very small molecule things that determine the rates, the functionality, the health of entire pathways. And they are, again, what is needed to conduct enzymatic reactions, and they're also the product of these reactions. So we get a very, again, sensitive look at the metabolism. And they're reflective of everything: lifestyle factors, disease, environment. I asked Robert today if he was fasting, because a postprandial metabolomics profile in the same individual looks vastly different than one that is taken with at least an 8-hour fast. So the idea is to provide our customers with a snapshot of their health. So we're going to talk a little bit about one of our studies and how this is kind of conceptually done. We will get more into the science as we go along. This is just to sort of get our feet wet. This is a study looking at 4 groups of Alzheimer's post-mortem. We're looking at frontal gyrus homogenate post-mortem from people who were no evidence of disease, people who had high pathology but had no symptomology of Alzheimer's, those who were classified as mild cognitive impairment that was consistent with aging, and then we had the confirmed Alzheimer's with biopsy and pathological examination. This is the general flow of a metabolomics study, where we take our study, we take 48 individuals, or however much we got from the biobank, we stratify them based upon their demographic data. That's me doing the work in grad school. We put them on various analyses depending on what markers we want to look at. So for this, we did an untargeted GCMS profile, and then we did a targeted LCMS profile. We collect the data off the mass spec. We then go through statistical analysis. We identify some biomarkers. But the goal is to identify the metabolic networks that are then causing amyloid beta deposition, tau tangle proteins in a healthy brain versus an Alzheimer's brain. Can we identify pathway level effects? Because disease doesn't happen, we'll see later on, disease does not happen at a metabolite level. It happens at a pathway level. So when we find these biomarkers, what we're saying is in that the preponderance of individuals in this study had elevated levels of, let's say, or decreased levels of palmitic acid, okay? That doesn't mean everybody in that study is going to have decreased levels of palmitic acid. It's a gross measure based upon the average of those individuals. However, everybody in this study is going to have dysregulation across a pathway. It might be this metabolite in that pathway, it might be another metabolite, but combining these through time, we are getting at multivariate analysis. We're going beyond the interpretation of a single metabolite. So we'll see this in the lab in just a minute, so I won't spend too much time on it. This is the platform we use to run samples. This is Agilent GCMS. We'll go through our metabolite list, and we'll go through how this works. But it's a very sensitive and specific way to look at the data, right? We get specificity because we do gas chromatography coupled with mass spec, and then the sensitivity comes from the mass spec itself. So mass spec is able to count the molecules one by one during a certain time that we set it to. Oh, we have a question? No. So we produce a metabolomic profile, and this is sort of your digital twin. We reconstruct an individual based upon, and this is a truncated version. Obviously, you can see it goes from 50 to 41, 50 to 81. And so the full panel we'll discuss, of course, when we talk about the report and go into the interpretation of it. But this is what we're trying to bring to people, is a full metabolomic profile that is not available. And then we generate that digital twin, and this enables doctors like yourselves, researchers like me, to visualize those complex processes in silico, offer a platform where we can simulate various versions of this intervention. So we'll take like niacinamide, and we will simulate niacinamide in various forms: ribonucleoside, adenine dinucleotide, the mononucleotide version. We'll simulate it at different dosages. We'll simulate at different times of day, breaking apart the dosages. We'll get into that in the case studies and how that's really done and visualized. But we can do this very no-risk simulation in a digital sandbox, get everything right, and then leverage the results of those for your patient in vivo. And then, of course, it's a prediction tool. And as it learns an individual, the predictions become more accurate. As it refines its delta parameter, or the difference between how we predicted this individual will respond and how they actually responded, this becomes more predictive over time. And as it becomes more predictive, it becomes an engineering tool. We're not only trying to predict where they'll be, but actually get them there through the interventions that we recommend. So these are some very sort of superficial case studies. But for instance, we have this individual, and we'll get into more detailed case studies with the actual data blown up and analyzed. So one thing that we saw was, Anita. Anita was sort of a friend of the company. She struggled with persistent fatigue. She was a breast cancer survivor. This was obviously the effects of chemotherapy. And so this is something that comes along with it. But Anita felt very debilitated, and really she couldn't make it past noon without having to sit down. And she was going through these behavioral protocols where she's pacing herself throughout the day so she can get things done without being completely spent. And what we looked at was basically mitochondrial markers, and her mitochondrial markers were a little bit diminished. What we saw was a deterioration of mitochondrial DNA due to oxidative stress. And so we were able to offer her mitochondrial therapies. I think she got high antioxidant diet and alpha lipoic acid, which is itself a very potent mitochondrial antioxidant. And then, of course, over the subsequent months she retested, she reported that she was back to her pre-chemotherapy vitality. We had Jake, unrelenting fatigue, cognitive difficulties. Oftentimes in the functional space, they'll say chronic fatigue syndrome, fibromyalgia, whatever crap else they can't figure out. And it's a metabolic issue, right? So we can look at this and we can say, okay, so again, we have some issues in purine metabolism. So we have some issues related to, DNA, RNA signaling. And then we also have this oxidative stress marker. So we didn't actually know this answer, but Jake's physician who ordered the test, and Jake is a pseudonym, of course, recognized this as a really rare inborn error of metabolism, one that's not screened at birth in the United States. So changed the diet up a little bit to include higher fats, and gave him some enzyme supplements, which we weren't a part of. But we were able to provide this physician, in this case it wasn't an in-house interpretation, but we were able to provide the physician with the data they needed to figure out something that our model couldn't. And then we have Rohan, persistent respiratory infections through childhood. Always used to get them at the same time of year too, so it was kind of chalked up to changing seasons. But we saw issues with his lipid metabolism. We saw certain fatty acids that were essentially triggering a chronic immune response. Specifically, we saw aberrations in palmitic acid, which is an aryl hydrocarbon receptor agonist. So his physician, was working with an immunologist, gave him some immunoglobulin therapies, changed the diet according to what the protocol had instructed, and Rohan's symptoms cleared up, and he hasn't reported any more of these seasonal respiratory infections since. So again, yeah. I mean, is it allergies, or is it some sort of systemic immune response to something that is in their metabolism that is not being identified? So basically, what are we trying to do here? I mean, we've talked about this again and again and again. We want to customize therapies. We want to do very specific, precise dosing. We want patients engaged in the process of reading their report, talking with our consultants, or with you if you choose to consult with them first, and staying proactive. So once we fix individuals, it's really an endeavor of maintenance, right? And Medicine 3.0 is a proactive endeavor. It's not reactive. And then data-driven, right? We have to trust the data. We were talking before the session started about a physician who didn't trust the succinic acid levels, right? And I quoted a Nobel laureate, Jack Arthur Dempsey, who is a fundamental figure in mass spectrometry, and he won the Nobel Prize in the late 30s for some mass spectrometry developments. But he said the mass spec does not lie. If you do not understand something, do more thinking. And so we are using an incredibly well-validated and rehearsed platform and method for collecting this data. And so the insights have to be data-driven, and they have to be accountable. So how do we do this? Well, in the study that we looked at, the metabolic profiling, what we found was, okay, first of all, we can take relative abundances of these 4 metabolites when we do an ANOVA test. So we look at the analysis of variance. We see these are all significant, right? In fact, the LSDP, so the least significant difference, it's a controlled significance statistic, is less than 0.05. So less than a 5% chance that these are due to, less than a 5% rate that this is due to type 1 error, or just a chance. That if we were to sample an external population, would we get the same pattern of changes in these metabolites? So yes, we would. 95% confident we would. So we have these 4 metabolites that can clearly tell between normal controls, high pathology controls, MCI, and AD. So we have biomarkers that can tell us about the pathology that's developing and the symptomology that's developing at each stage, right? So there's some translational effort that has to be done. Keep in mind this is post-mortem neocortical tissue. You can't do this in living people, but we then go to the serum. We do another trial, right? We keep validating this in the translational pipeline inches forward. When we do a random forest classification, so this is a machine learning method whereby decision trees are made. We make 500 decision trees, and for every iteration, we either see the out-of-bag error go up or down. And what we can see here is that we have an overall trend line, which is kind of useless in this case, but we can clearly delineate between all 4 of these at almost 220 decision trees are what's needed in this model to delineate between normal control, high pathology control, mild cognitive impairment, and AD. So 4 metabolites, 200-something decision trees. You can run it all the way to 500. It won't change significantly. And when we look at the area under curve, so when we do a receiver operating characteristic analysis to test the sensitivity and specificity, what we see is that we have a 91.7% accuracy. So when we do leave one out cross-validation, so we did 100-fold leave one out cross-validations. What that means is an internal permutation test, whereby we take the 48 people, we leave one out, we test the 47. Then we put that one back in, leave another one out, test the 47. And we try to test the accuracy of every iteration. So we do that 100 times, and we see with the random forest classifier, we get roughly 92% accuracy. Very sensitive, very specific method of doing this. Of course, again, this is post-mortem. So what about when we look at inflammation? There's another publication. This one came out in Acta Neuropathologica. Parts of this data is actually in our model. So this is one of our really higher end publications. Acta Neuropathologica is like an impact factor of like 15 or something, very good journal. And what we did here was in mice and in humans, we looked at choline. So we had triple transgenic mice. I'll stand over here. We had triple transgenic mice and non-triple transgenic mice. So these are mice that have preponderance to develop Alzheimer's. They've been genetically edited with a couple of knockouts, and if left to their own devices, they develop Alzheimer's. And when we test their behavioral mechanisms, either it's in a maze or a swim test, they fail, they die, they have Alzheimer's, right? So we validate these models. We put some on a choline diet, some on a choline deficient diet. We looked at the plasma and the cortex of these individuals, and of course we see things that we'd expect. So even on a choline deficient diet, the triple transgenic mice, the ones that are engineered to develop Alzheimer's, have significantly lower levels than their counterparts who are already on a non-choline diet. So the choline deficiency doesn't matter. If you are a triple transgenic mouse in the study, you will have lower choline deficiency. You'll have lower choline levels even compared to a choline deficient C56 black mouse. So we see cortex choline is down, acetylcholine is down, TNF alpha is up. Cortex TNF alpha is up. We see plasma choline is significantly associated, as we would expect, with cortex choline. This is measured in micromoles per liter. Same thing with acetylcholine, TNF alpha, and this is for the non-transgenic and the triple transgenic mice. And then over here is we see these metabolites, and these are what is baked into our analysis as well. So what we see in humans, so this was in mice, and this is in humans, we see that we have, and this is serum choline and acetylcholine, the same, and this is in the same study. So we did mice and we did humans in the same study to show sort of multimodal effects. We see that individuals who had moderate Alzheimer's to severe Alzheimer's were basically deficient in choline, and then we see people who were in the control group had very high levels of acetylcholine. And then when we look at these 4 metabolites, we see that they're incredibly predictive as a whole for their choline status. So we're looking at resilience to Alzheimer's from these metabolomics. So when we take your test, right, Gail, when you get some people back and we see, okay, phosphatidylcholine is the recommendation, or choline maybe sometimes we recommend. It's based on a lot of these analyses that we've done and some other studies that this research is built on. So for instance, we were talking about, we look at pathway effects. This is a really good example of why we look at pathway effects. When we look at things like valine, or we look at ferulic acid, it should generally be higher in the control group. But we see this one person that's very low. So if we were only looking at ferulic acid, we would look at this person and we'd say, you know, I think they're actually pretty good in their acetylcholine status. They should be over here. But once we get to look at their entire profile across these rows, now we're like, they're somewhere between moderate and severe. So this is a really good visualization via the heat map that why we don't use single models, single univariate interpretation, because univariate interpretation can be wrong. It's from frequentist statistics, which have grouped people and averaged them, and the average is not the person, right? That's not precise. So by looking at more than one variable, we get to have very granular interpretations of that. So here's a real quick, we're a little bit behind, but that's okay. The lab tour really shouldn't take a whole hour. We're going to do a lab tour on the 3rd floor right after this, and then Alex is going to prime you on systems biology and go through, again, the concepts and the hierarchy and interrelations. We're going to break for lunch at 12, and then we're going to go at 1. We're going to meet back in this room where we're going to talk a little bit about tests and diagnostics in precision medicine. Yeah, and so we're gonna do that, and then we're gonna talk a little bit about, in this talk, I'll talk more about the test itself, and what we're seeing and what we can do with it. And then we'll go into another talk, which I hope you enjoy. It's a very sort of like deep science. I'm gonna invite you into the deep end of the pool. You're all brilliant healthcare providers, so I imagine you can tread the water. And then we're gonna go really into early detection of breast cancer with IVDs and showcase some papers there. And then Gail, she's going to give her talk on how she customizes the protocols that we give based upon the treatments that she has in store and, of course, her own vast experience as a physician. And then we'll have a little Q&A panel back in this room. And then tomorrow we're going to start at 9. Gail's going to kick us off with how she integrates this test into her practice, both from a medicine perspective as well as the financial side. And then Justin Lesh will be joining us to talk a little bit about business success. I know nothing about that. And then we're going to do a report interpretation and translation, whereby we'll hand out some reports, we'll talk about it, and we'll see what you guys think and what patterns that we can glean from what the AI is reporting.